So this was the last slide from part 2 PowerPoint for adaptive, and we didn't really go through it, it actually probably is a good summary introduction to the continuation of the story today. So antigen presenting cells, which we'll talk about, right, are needed for no matter what type of T cell you're talking about, right? And so this molecule that antigen presenting cells use to present antigen is called what? It's abbreviated, right? MHC, which stands for Major Histocompatibility Complex. And there are 2 of them, right? We have class 1, which is found on all nucleated cells. This cell has a nucleus. So could that be class 1? Absolutely. Could it be class 2? Sure, right? We're going to figure out in a minute which one it is, right? Because we're going to see what happens. So, this cell is presenting the antigen. The antigen, right, the foreign substance is this little yellow thing. The T cell has a T cell receptor that's recognizing it, right? And this co-receptor is a CD something, right? Which if that was labeled 2, it would tell us what MHC this is, right? And neither one of those is labeled in this picture. It's probably a good thing. I could put this on the test even and ask you which ones it is. Okay? Because there's a clue further down that's going to help us figure out. So this is an effector T cell. Notice it's got vesicles inside of it that contain toxic substances. It says cytotoxic killing, right? So notice it's releasing these toxic substances on a cell, right, that's infected with clearly that look like viruses, right? So if it's, say, any one of our cells, maybe say a skin cell, right? Which MHC would it have? 1 or 2? 1. Remember, 1's the most prevalent, it's the most important, right? So this is MHC class 1. This is a cytotoxic T cell. Cytotoxic T cells have CD8 because they eat, kill, right, infected cells. So there's some clues here that help us figure out, even though these things weren't labeled, what they are. Okay? In this activation here, right, some of these activated, now we know it's a cytotoxic T cell, right, become effector T cells, cytotoxic T cells that hunt and kill infected cells. Some of them, right, because we're going to clonally expand now, right, something we're going to talk about today. We're going to make multiple copies once this is activated, right, and this antigen presenting cell is going to activate it. We're going to produce memory cells that stick around. So that way, if, right, the bad guy comes back again, an antigen presenting cell comes, it could activate a memory cell instead. This makes the process go much faster. Same thing's going to happen. Notice feedback to here, right? We're going to get effector cells, hunt and kill infected cells, and more memory cells produced. Every time, right, we see it again. So that was the end of 2. We're going to start 3 today, which kind of reviews some of that stuff. So in order to get this process started, right, we have to have some way of that presentation that you saw happening in the very first picture in that flow chart. So cells can, especially phagocytic cells, do phagocytosis, where they literally take large things like bacteria and viruses and they engulf them and, remember, destroy them by phagocytosis. Some things like viruses get into our cells, right? And they'll get into endosomes and sometimes those can be processed by the cell, and that stuff will get presented. Sometimes cells are going to surround molecules, right, like proteins, and they'll surround them in vesicles. This is called penocytosis, sometimes referred to as drinking. Phagocytosis is sometimes referred to as eating because they're much larger. Irrespective, the stuff is either we're taking it in or it's getting in to our cells. So stuff that typically stays outside of our cell, like bacteria, most bacteria are not intracellular bacteria. They grow outside of our cells, right? Viruses, on the other hand, we know have to get in. But proteins, toxins, things like that, you know, even the viruses themselves have a period of time when they're outside of our cells, right? So if it never gets inside our cells, we have no way of processing and presenting it to T cells. So exogenous means outside, right? How are they processed? Right? They're extracellular antigens from outside the cells. We're going to take them in by those pictures that you saw, right? Phagocytic cells will engulf whole bacteria, right? Some, especially viruses, will bind to our receptors and we'll take them in. This is how they get in. But this is actually good for us too, because then we can bring them inside and process them sometimes. Or we're going to take in large proteins, right, by penocytosis. The important thing is somehow, right, it gets into a cell, and hopefully one of our specialist or professional antigen presenting cells engulfs it. So do you guys remember who our professionals are at this? Who's their main job to do phagocytosis? Neutrophils, but neutrophils just eat and kill, right? That's it. Macrophages, on the other hand, which are your big eaters, are going to do that. Dendritic cells, which we haven't talked much about yet, but we're going to talk about, they're the main ones that are really important to this process of antigen presentation, especially when we're talking about a naive T cell. Not a memory T cell, but a naive T cell that's never seen antigen before. Because remember, we make these ahead of time. We talked about that last time. And then we're going to introduce today, B cells have this ability to, not so much for activation of T cells, but activation of themselves. Okay? B cells are the last thing we talk about. They produce antibodies. But as you can see here, right, they're going to bring it in, they're going to process it, they're going to load the foreign substances onto MHC, in this case class 2, and they're going to present them on the surface so that a T cell can come along, right, and inspect it. So if this is MHC class 2, which T cell is it restricted to talking to? There's only one T cell. We have two types, right? We have CD4, which we also call helper T cells, and we have CD8, which are called cytotoxic T cells. So for MHC class 2, anyone know which one it is? It's the helper T cells. And I just, sing the song 2, 4, 6, 8. Who do we appreciate? And I usually remember that MHC class 2 goes with CD4. Whatever works. But pretty much, right, MHC class 2 is also restricted to those antigen presenting cells. They're the only ones that talk to helper T cells. Right? As another clue for you. So CD4, right, that co-receptor binds to MHC class 2, right? So how, right, are antigen presenting cells interacting? It literally is a receptor to receptor and co-receptors, right, interactions. What's the result though? So, okay, this T cell's receptor recognizes this foreign antigen that this antigen presenting cell, right, dendritic cell, macrophage, or maybe B cell is presenting, right? They know they can do this handshake because CD4 recognizes MHC class 2. Okay. So what? What's going to happen next? This T cell, this one T cell, is recognizing this antigen. We call this activation, right? When the recognition is happening. And then there's cytokines and other co-receptors not shown here. This is the main interaction. Do we want just one helper T cell? No. It's going to go into proliferation, right? So we have a whole bunch of these T cells, helper T cells, that know this information. And helper T cells mostly are going to do what? These are your talkative cells, right? They help out. How do they help? They talk to all your other cells. How do they talk? It's a chemical interaction, right? Cytokines. They release a whole bunch of cytokines and talk to the different cells to tell them what to do, right? So this interaction, right? So when they take something in from outside, this is restricted to, as we saw, helper T cells, CD4, MHC class 2. Once it's initiated, the helper T cell is going to become activated. We're going to get a whole bunch of them. Some of them are going to become effector cells, which produce large amounts of cytokines, lots of talking. Others are going to become memory cells, in case the bad guy comes back again later. So I like these cells because, you know, I can relate to them. So here's a dendritic cell, which is a professional antigen presenting cell. It's presenting via MHC class 2, right? So it's taking in this foreign substance, right, from the outside. It's presenting an MHC class 2. It's talking to this helper T cell, this CD4. This CD4 recognizes it, so it becomes activated. So one of the things that it does, right, that signal is going to get sent to the nucleus. It's going to produce more receptors for IL-2. And it's going to make the cytokine IL-2. Remember, IL stands for interleukin. Look at what's happening here, y'all. Not only is it releasing IL-2, but the IL-2 it's releasing is binding to its own receptors it's also making. This is called autostimulation. They literally talk to themselves. See why I can relate? I have moments of that, right? And so then, again, they're going to proliferate. So we're not going to have just one of these. This says, IL-2 says proliferate, right? Make more of yourself. So it's going to clone itself so that it has all its clones have the same receptor, right? That same T cell receptor. Some of them are going to become effector cells that are going to produce more cytokines, talk to other cells depending on what happened here in the beginning. What did they get exposed to? And then memory cells that'll hang out in your lymph nodes, right? Your spleen, your lymphoid associated tissue, different areas in case the bad guys come back. So what does IL-2 do? Right? It auto-stimulates helper T cells to proliferate. It talks to itself. So endogenous antigens, how are they processed? So endogenous means what? Inside. Endo means inside, right? So these are intercellular pathogens within the cell. So this is when the virus gets inside the cell, right? When the intercellular bacteria gets inside the cell. We have a special proteasome, right? So this is a set of proteins. They're proteolytic. That when ubiquitin, another protein, binds to foreign proteins inside of our cells, it'll process them and break them up into little pieces. This is necessary so they can be loaded onto MHC class 1. This happens in all your cells, right, that have MHC class 1. But only your professional antigen presenting cells are actually going to be able to talk to the T cells, right? So this happens, right? Proteins, right? They get processed. They get sent to the endoplasmic reticulum where they get loaded onto MHC class 1. They get processed and sent through the Golgi apparatus, which says you've got to go to the cell membrane surface, right? And so which T cell is MHC1 restricted to, talking to? The cytotoxic T cells, right? CD8. Because remember, if this cell is infected on the inside, what do we want eventually to happen to it? Death, right? So, close up, right? What is the result here? Okay, so we have, again, a dendritic cell or macrophage is presenting a foreign peptide to a cytotoxic T cell. CD8 recognizes MHC class 1. That's their secret handshake. But most importantly is this T cell receptor is recognizing this foreign antigen, this peptide. So that's going to cause this T cell to become activated, right? So we're going to have more than just one cytotoxic T cell. Except, guess what? Cytotoxic T cells cannot talk to themselves. They're pretty powerful cells, right? They go out and kill other cells. We kind of want to make sure that we really want them to do that. So, although part of the activation is this interaction with the antigen presenting cell, that macrophage or dendritic cell that process that endogenous antigen, the helper T cells are also going to be activated, not shown here in this picture. They're going to release IL-2, which we know, right, is going to cause the cells to proliferate. That's what is going to happen before you're going to actually get proliferation of a cytotoxic T cell. So one extra step here. Right? Cytotoxic T cells cannot auto-stimulate themselves to proliferate like helper T cells can. So at some point, right, a helper T cell also has to get activated along the same time as a cytotoxic T cell. Something we'll talk about later. But same result, right? We're going to get effector cells that hunt and kill infected cells, effector cells, and we're going to get memory cells that stick around. Right? So how do they kill? Right? We saw this already. Right? They will bind to infected cells. In this case, look, it's a bacteria on the inside. Right? But we're picking up little pieces of that bacteria. We're presenting it. Right? This already activated cytotoxic T cell recognizes it. It's going to release granzymes and perforin. Perforin, as the name implies, perforates the membrane of that cell that's infected. Granzymes will digest proteins. It will cause this cell go into programmed cell death, right? Apoptosis. What's important about programmed cell death as opposed to other types of cell death? What does it not elicit? Inflammation, right? We don't get an inflammatory response typically with this. So activation of T cells, right? Effector killer T cells. That antigen, that little piece is the epitope, and they're going to kill, right? And induce programmed cell death, apoptosis, without inflammation. So this is one that, if you'll note, I put a note not on exam this year. But think about this, right? What does cross-presentation mean? It means you're going to present both ways, right? Both MHC class 1 and 2. Dendritic cells or macrophages, sometimes when they take things in, especially viral antigens, think about this. They're outside of our cells sometimes, and viruses get inside of our cells. So we need both types of protection, right? So these cells can do what's called cross-presentation, right? Where they're going to present not only MHC class 1, but 2. So that we get, right, both cytotoxic T cells and helper T cells presented. Especially if the virus doesn't infect a professional antigen-presenting cell, right? We really need this process called cross-presentation, right? So, where they do both, right? Present both ways.
I have a question, Ms. Burns.
Mm-hmm.
Is MHC1 exclusively endogenous? I mean, that seems very...
Yes and no. Endogenous means comes from within the cell, right?
So like a virus?
Like a virus, right? But if a virus doesn't get into an antigen-presenting cell, we can't present it on MHC class 1, right? But depending on what it is, your dendritic cells and your macrophages can do both, right? They can present exogenous antigens, right, with an internal stimulus, right? It gets real complicated, right? Which is why we're going to skip over it, right? And just know, right, that these cells have the ability to do both, depending on what gets in, right? Right? So, what's the purpose, right? Proteins from one are shuttled to the other so that we can get this presentation. So we can get an intercellular pathogen such as viruses that do not infect any type of antigen presenting cell too, right? So it kind of covers all our bases. All right, so this is from those web pages, right, which the link is not working anymore, but I found a copy, right? So these are cute, right? But they don't go in quite the detail we need. So again, we can do a little bit of, right, applying our knowledge, right? So here they tell you this is a dendritic cell. And we're going to talk about him soon, right? So here is probably a bacteria, right? And he's wearing this cute little crown, right? And he takes it in and he processes it and he presents it on the surface, right? This piece of that bacteria. You see that? But what they're not showing you is which MHC would it be presenting? Well, dendritic cells have both, right? So we got to go to our next clue, which is they say this is a helper T cell, right? So that would be which one? CD4 or CD8? This would be CD4. And we know CD4 is restricted to MHC class 2, right? So it's class 2. What cells have this? Dendritic cells have this, right? What does MHC class 2 do? It actually presents the antigen. So that receptor is not shown in this picture, right? It's not very detailed. You do see a T cell receptor, right? You see this happy afterwards activated T cell. But we know since they say this is a helper T cell that it's CD4, right? What cell has it? Helper T cells, right? Only. What does CD4 do? This helps us recognize MHC class 2, right? Again, not shown here. But this interaction, right, this guy is activating that helper T cell. So he's going to proliferate, right? And he's a big talker. I mean, I guess the big mouth is appropriate, right? Because he's going to do a whole lot of talking. So that's the end of